Hi, I'm Matt Spear, Chief Medical Officer of Poseida Therapeutics, a cell and gene therapy company located in San Diego, California. Here are my disclaimers. So as I mentioned, Poseida is a cell and gene therapy company that was spun out of Poseida Biopharmaceuticals approximately 5 years ago. We primarily focused on CAR T cells since then, but now moving back into straightforward gene therapy paradigms, a correction of monogenic diseases. Obtained significant funding over the years, now have approximately 170 employees. You can see our leadership team on the right-hand side of this slide. Our company focuses on two primary platforms, one for gene insertion, that's the piggyback transposon system, and one for gene editing, Cas-Clover system. We also have developed several different supportive platforms around these to increase the capability of these platforms to achieve the goals intended. First, let's talk about the piggyback transposon system. Transposons are plasmids which carry a cargo, i.e. the gene of interest, to be inserted into a cell, flanked by two ITRs. They, i.e. the plasmids, along with a transposase, are transfected into a cell. So this is a non-viral transfection or transduction system. The transposase cuts the ITRs out of the plasmid, the source plasmid in the cell, and inserts it into the host genome. So this has several advantages. Not only is it non-viral, but it has very long, large cargo capacity because it is not limited by viral-based cargo capacities. Thus, we could put multiple or many genes into a cell. And subsequently have found that this preferentially favors transposition, i.e. transduction of TSCM cells, which we'll talk about in a little bit, but have been covered in several different publications over the years as a preferable stem cell or preferable cell for CAR T-based therapies. The other major platform is the Cas-Clover gene editing system. This is effectively a combination of CRISPR and zinc finger or TALEN technology, where it uses two guide RNAs, a left and a right guide RNA, to bring together the CLO51 endonuclease cutter to achieve the editing. Because of this, because of the two guide RNAs, it has effectively no, i.e. we have never detected off-target cutting. It can also be used in stem cell memory T cells, particularly useful for our CAR T cell platform. We're also using not only standard AAVs to do gene transfer with transposons, as well as the electroporation methodology I talked about earlier, ex vivo, but also developing new nanoparticle technologies to allow in vivo gene transfer without, or potentially without, the toxicities, manufacturing delays, and costs of AAV technology. This is our current pipeline. Once again, we started with autologous CAR T cells approximately 5 years ago. We first developed a BCMA-targeted CAR T cell, PBCMA-101, for multiple myeloma. Subsequently moved into solid tumors with PPSMA-101, targeting PSMA for castrate-resistant prostate cancer. We're now moving into allogeneic CAR T cells, finally utilizing our Cas-Clover editing system for a clinical product, as well as moving back into the treatment of monogenic diseases, inborn errors of metabolism, such as ornithine transcarbamylase deficiency, i.e. POTC-101, our latest product. Right. Let's talk about our Immuno-Oncology CAR T cell programs. The piggyback transposon system and the Cas-Clover gene editing system allow for a number of advantages in the production of CAR T cells. First of all, with autologous CAR T cells and the TSCM product preferentially produced, there's safety advantages as well as potential efficacy advantages. From the standpoint of the Cas-CLIPper gene editing system, we have the ability to produce fully allogeneic CAR T cells and all the accompanying advantages of an allogeneic product in terms of being off the shelf and ready to use. Let's talk a little more about TSCM. TSCM are the stem cells for T cells. So they are the long-lived progenitors of effector T cells, which actually kill pathogens and/or tumor cells in the case of CAR T cells. They are the same cells that produce long-lived immunity to a pathogen when a vaccine is administered. So you can see the potential advantages that these cells would remain around for a long period of time, potentially carrying out anti-tumor activities over that period of time, as well as slowly or gradually expanding to produce effector cells, which may lower the rate of CRS, cytokine release syndrome, associated with frequently CAR T cells. So these are less differentiated cells, preferentially produced by piggyback transposons. And they have the benefits, or potentially confer the benefits I just spoke about. We tested our first product, PBCMA-101, a clinical trial over the last several years. And you can see that on the right-hand side of this graph, significant response rates in patients with very low CRS rates, as you see on the left-hand side of this slide. Next, we're moving into allogeneic CAR T cells. So using the Cas-CLIPper gene editing system, we are removing, i.e. knocking out MHC class I and the TCR complex, thereby potentially allowing these T cells to be administered to multiple patients from a single donor, meaning effectively off the shelf, and potentially with all the advantages we see with our autologous CAR T cell products. Interestingly enough, we've also used one of our supplementary platforms to overcome one of the significant issues with producing allogeneic CAR T cells. That is, if you are knocking up the TCR complex, you lose much of the ability to stimulate these cells to replicate and produce larger quantities of CAR T cells during manufacturing. We have developed a proprietary booster system which overcomes this and allows these CAR T cells to multiply significantly during manufacturing. You can see here they have produced significant numbers of doses from single donors, effectively single leukopaks of T cells. And you can see on the right-hand side, we've preserved the TSCM component of this using the piggyback transposon system. We've tested this in a murine model of multiple myeloma. This is our PBCMA Allo-1 product, which targets BCMA. And you can see that the efficacy in this preclinical model, this murine model, is as good, if not better than our autologous product, as well as others that you would see presented in the literature. Let's talk about solid tumors now for a few minutes. You can imagine where a TSCM product could have significant advantages in the treatment of solid tumors with CAR T cells, in that one dose could produce waves of CAR T cells, which could continually eat into a solid tumor. We have utilized our PBCMA-101 product in murine models. Recently, we've started a clinical trial. You can see that in the murine models, this completely eliminates an Allen-Cap tumor model, i.e. a prostate cancer tumor model, in mice with both low doses and high doses of CAR T cells, effectively equally as well, supporting the hypothesis I just mentioned. Moving on to some very interesting findings during the conduct of these experiments, you'll note that the CAR T cells, which are TSCM phenotype preferentially, noted in the upper right-hand quadrant of these FACS plots, start out in both cases, top and bottom row, with a mock and a tumor transplanted mouse. But in the tumor transplanted mouse, the T cells differentiate into effectors, then redifferentiate into TSCM cells, potentially conferring longer-term tumor resistance. The next target we are working on, relevant to a broad area of solid tumors, is MUC1C, a novel tumor-associated antigen. In particular, we're looking at a novel target within this tumor-associated antigen. You can see that MUC1 is expressed on a broad array of epithelial-derived tumors, the left-hand graph. The majority of patients with breast cancer, prostate cancer, non-small cell lung cancer express this antigen. You can see down below that when we create allogeneic CAR T cells targeting MUC1C, we confer them with cytotoxicity against the broad array of tumor types. We've also tested this in animals, and you can see that in both a triple-negative breast cancer model and ovarian cancer model, P-MUC1C allo-1 eliminates tumors. One of the other advantages of the piggyback transposon system is the ability to carry multiple genes. So we've tested this out with CAR T cells in terms of adding sequentially up to 4 different binding motifs to target 4 different tumor-associated antigens. And you can see in these figures here that as we add 1, 2, 3, 4 CARs, chimeric antigen receptors, to our CAR T cells, they develop the ability to target and kill cells expressing each one of those antigens. We're in the process of translating this into potential clinical products, i.e. multi-CAR T cells, targeting a significant number of relevant antigens. Let's move on to our gene therapy programs. As we mentioned, the transposon system, generally used ex vivo to produce CAR T cells, permanently transduces, i.e. transposes, the target cells. We can also utilize adeno-associated virus or other transduction mechanisms to insert transposons and transposase in vivo, i.e. in vivo gene therapy in a living animal. So a large number of indications, monogenic diseases, inborn errors in metabolism that could be corrected with this methodology. The company has initially focused on ornithine transcarbamylase deficiency and has preclinical data created years ago with the product that indicates that animals can be treated successfully. We've now moved towards models of methylmalonic acidemia, another very severe inborn error of metabolism. So you can see here that we can inject AAV carrying a transposon along with AAV carrying a transposase into mice and transpose, i.e. transduce the majority of liver cells in those animals. If, as you see on the left-hand side, you don't include the transposase, the amount of transduction is much less, indicating the importance of integration and the transposon system versus utilizing an adeno-associated virus system alone. Here in this model, we have demonstrated proof of concept in a murine model of ornithine transcarbamylase deficiency, i.e. OTC, and that the POTC 101 product corrected ornithine transcarbamylase activity levels in mice and resulted in survival of those mice with corrected OTC. We're now moving towards non-viral technologies here as well. So we've developed proprietary nanoparticle systems for moving both the transposase and transposons into animals. And you can see here that murine livers have been transposed with luciferase successfully using this technology. Thank you again for hearing more about Poseida's